Definitions of DNA technology. Recombinant DNA technology is a series of techniques used to manipulate and isolate DNA segment of interest. In order to insert this isolated DNA into a cell for cloning, some type of factor is commonly used. To create such a recombinant factor of bacteria containing of plasmid and cell containing of DNA of interest is used. Plasmid from the bacteria is combined with the gene of interest that has been isolated. This plasmid is separated with a circular DNA located in the bacteria, and when we look close at it, we can see that it consists of complementary DNA strands just like normal DNA. By using restriction enzyme to cut the circular DNA up to a specific point, one can create two sticky ends that are complementary to the DNA of interest, allowing for much easier binding of the gene of interest to the plasmid. Then, once the DNA strands of gene interest are bound to the two sticky end plasmid DNA, ligase can be used to ensure proper binding. What are we left with is a recombinant DNA plasmid, which can be inserted back into a bacterial cell for cloning or into mammalian cell for transfection.
Hi, we are going to talk about tools used in recombinant DNA technology. So, for the first one is target DNA. Target DNA is a desired gene from other organism or species to be inserted into a selected DNA cloning vector. The second tool is DNA cloning vectors. DNA cloning vectors, such as plasmid, are one of DNA molecules that can carry foreign DNAs and to be inserted and replicate in the host cell. So, what is plasmid? Plasmid is a small circular DNA bacteria. It can replicate with or independent of bacterial DNA. It genes function like normal genes. It provides traits like antibiotic resistance and also can be modified and reinserted into bacteria. For your information, there are three components contained in plasmid. The first one is ORI, origin of replication, where it binding site of DNA polymerase to allow replication on its own. The second one is multiple cloning site, MCS. It has various restriction sites where the restriction enzyme cut. And for the last component is an ampicillin, antibiotic resistant gene, AMPR, where it is a selectable marker gene. So, what is the characteristic of plasmid as a DNA cloning vector? First, plasmid must evolve to accept foreign DNA at multiple cloning site, MCS. So, the cloning vector must have MCS for cloning. The second one, the plasmid evolved to replicate independently in the host cell. So, the cloning vector has origin or replication. And the third, it has selectable marker gene, where marker gene code for antibiotic resistance. Alright, next is restriction enzyme. Restriction enzyme, also known as endonucleases. It can recognize a restriction site and cutting both DNA strands. Most restriction enzymes cut palindromic sequences. So, what is restriction site? Restriction site is DNA sequence containing the cleavage site that is cut by a particular restriction enzyme. Now, what is palindromic sequences? Palindromic sequences is the base sequence of one strand read the same as its complementary strand when both strands are read in the 5 to 3 direction. We have two types of restriction enzymes. The first one is sticky end, where it have complementary overhangs, and the other one is blunt end, where it have uniform edges and no overhangs. Now, for the fourth tools, we have modifying enzymes. Modifying enzymes consist of two enzymes. First, DNA ligase. DNA ligase used to join fragments of DNA together, while Taq polymerase is used to replace DNA polymerase in polymerase chain reaction (PCR). Now, for the last tools used in recombinant DNA technology, host cell. The cells that receive, replicate, and express the recombinant DNA within cells. In order to replicate, cloning vectors carrying the target gene must be introduced into the host cell. Host cell is cell which able to accept foreign DNA that is the cloning vectors and allows the vectors to multiply. Organism commonly used is bacteria such as E. coli. The process of introducing recombinant plasmid into host cell is called transformation. Well, that's it.
Gene cloning is a technique used to produce clones with identical genetic makeup. Let's learn on how to produce transgenic bacteria that can form insulin. The target gene is cut from the chromosome using restriction enzyme. The DNA plasmid is isolated and cut using the same restriction enzyme to ensure that both the insulin gene and the plasmid have complementary sticky end. The target gene is then inserted into the plasmid using DNA ligase. The recombinant DNA is introduced to the host cell and allowed to be reproduced. Bacteria cell is then cultured in a medium containing antibiotic ampicillin and the sugar X scale. Bacteria with preferable markers is then cultured in large scale for commercial use.
PCR is a technique used to make multiple copies of a segment DNA of interest, generating a large amount of copies from a small initial sample. The tools required for PCR are thermocycler, target DNA, primers, nucleotide, and polymerase. The process of PCR involves the repetition of 3 steps, where the first one is denaturation, which separates the 2 nucleotide strands of DNA molecule. Next is annealing, which the primers bind to the single-stranded DNA. The last one is elongation, which 3 nucleotides are added to the primers in the 5' to 3' direction to form a double-stranded copy of the target DNA. The cycle is then repeated to produce a large amount of a specific DNA sequence.
Let's move on to the next subtopic. We're going to learn about the application of DNA recombinant technology in pharmaceuticals. For example, the mass production of insulin for diabetes patients. cDNA is a DNA strand made in vitro by using mRNA as a template and enzyme reverse transcriptase. cDNA has no introns and only contains extrons. The first step is to produce complementary DNA strands. mRNA that codes for the insulin is extracted from the beta-Langerhans cells from the pancreas. In order to use the mRNA as a template, first we must cut and remove the introns. Note that introns are non-coding segments, thus it is not needed here. And then we join the extrons together, which are coding segments, to produce a mature mRNA. After the transcription process is done, we will use the first enzyme involved in this process, called the reverse transcription enzyme. We will form a new single strand of cDNA, and followed by the degradation of mRNA by the second enzyme used in this process, called the mRNA degradation enzyme. In the final step, second DNA strand, which is complementary to the first strand, is synthesized by DNA polymerase. The double-strand cDNA is now ready to be used as a gene of interest in the next process of producing recombinant DNA. The double strand of cDNA we produced earlier is cut open using a restriction enzyme. In this case, we will be using BAMHI. This leads to the formation of 2 sticky ends on the cDNA strand. Meanwhile, a plasmid is isolated from the E. coli bacteria to be used as a cloning vector. The plasmid contains ampicillin-resistant gene and LAC-Z. The plasmid is cut using the same restriction enzyme to create complementary sticky ends on the plasmid. The cDNA is then inserted into the plasmid and joined using DNA ligase. This then produces a recombinant plasmid. Recombinant plasmids is then reintroduced inside E. coli that acts as a host cell through the process of transformation. The host cell reproduces and amplifies the amount of recombinant DNA. However, 3 variations of bacteria cells are possible: bacteria cells containing recombinant plasmids, bacteria cells containing non-recombinant plasmids, and also bacteria cells that does not contain any plasmids at all. The host cell is cultured inside a medium containing antibiotic ampicillin and sugar called X-gel. To distinguish bacteria cells from each other, bacteria cells with non-recombinant plasmids will form blue colonies, while bacteria containing recombinant plasmids will form white colonies, and bacteria cells that doesn't take up any recombinant plasmid wouldn't survive. The bacteria cells that produce white colonies express human insulin gene and produce hormone insulin are later further cultured in fermenters. The recombinant plasmids replicates independently of the bacteria chromosome. When plasmids divide, it is also copied to the daughter cells. As a result, many copies of the DNA encoding human insulin are produced. Finally, gene expression is induced and produce polypeptides. The polypeptides are then extracted and purified to produce functional human insulin. They also undergo further testing before use on patients.
Recombinant DNA technologies has significant application in agriculture, particularly in transgenic plants and transgenic animals. What are transgenic plants? Transgenic plants are plants that have been genetically engineered. Transgenic plants are developed through a breeding approach that utilizes recombinant DNA techniques to introduce new characteristics into plants. These plants are genetically engineered to possess desirable and heritable traits such as improved resistance to pests, tolerance to herbicides, or adaptation to environmental stress. For example, transgenic soybean, which has resistance to glyphosate herbicides. Next, for transgenic animals. Transgenic animals are animals that carries a foreign gene that has been deliberately inserted to its genome through genetic engineering techniques. This process allows scientists to introduce specific traits or study the function of genes, enabling advancement in research, medicine, and agriculture. Transgenic animals also led to development of fish and livestock with an altered genetic profile, for example, to grow faster, like salmon. Recombinant DNA also contributes in the application in environment. For example, to clean up oil spill by using Pseudomonas bacteria. These bacteria are naturally capable of degrading hydrocarbons, the primary component of crude oil. Using recombinant technology, scientists can enhance the efficiency of these bacteria by introducing specific genes that boost their oil-degrading capabilities. Another example is to control malaria and dengue. Recombinant DNA technologies offers innovative approach to combat vector-borne disease such as malaria and dengue by targeting mosquitoes population, the primary carriers of these diseases. Using genetic engineering, scientists can manipulate mosquitoes' genomes to reduce their ability to transmit pathogens or control their population.
Recombinant DNA technology has a lot of advantages. First, it grows faster. Bioengineered plants can grow at a faster rate and develop stronger, more resilient structure. They also require fewer chemical inputs such as herbicides, insecticides, fertilizers, and antibiotics, making them more efficient for agriculture. Secondly, it is higher nutritive content. Genetically modified GM foods are designed to have enhanced nutritional properties. This means they can contain higher levels of essential vitamins, minerals, and other beneficial nutrients compared to non-GM foods, helping to improve dietary health. Third, synthesis of pharmaceutical products. Transgenic organisms, which are genetically modified, can be used to produce valuable pharmaceutical products like drugs, vaccines, and hormones. These innovations are particularly beneficial for improving health outcomes, especially for people with limited access to medical resources. Fourth, it is also cheaper. GM foods are typically less expensive to produce compared to traditional farming methods, which can make food more affordable for consumers and reduce overall production costs. And lastly, it is also used in forensic science. Gene technology is frequently applied in forensic science to analyze DNA evidence. When used accurately, it helps law enforcement and the justice system to determine whether a suspect is guilty or innocent with greater precision. Now, we move on to the disadvantages in recombinant DNA technology. First, it might produce harmful byproducts. The process of creating recombinant DNA may generate toxic or harmful substances as unintended side effects. These byproducts could be dangerous to living organisms or the environment. Second, it may release GM organisms into the environment. If genetically modified GM organisms are accidentally released, they might spread and interact with wild species. This can disrupt ecosystems and harm biodiversity due to unintended consequences. Third, it also can cause cancer. Inserting new genes into a person's DNA could activate oncogenes, which are genes that have the potential to cause cancer. This poses a health risk. Fourth, it is expensive. The technology is costly because of the need for advanced research, equipment, regulatory approvals, and large-scale production. And lastly, it can lead to misuse of recombinant DNA technology. There is a risk of misuse, where someone might alter genes to create harmful traits, such as developing dangerous viruses or harmful organisms.
Surah Ar-Ra'd, verse 11, translate as, "Indeed, Allah will not change the conditions of people until they change what is in themselves. And when Allah intends harm for a people, there is no repelling it, and there is not for them beside Him any protector." This verse delivers two profound messages. One, change starts from within, which is Allah will not improve the state of people or a community unless they actively strive to improve themselves, whether in faith, character, or deeds. Second, Allah's decree is absolute. If Allah wills hardship as a result of wrongdoing or neglect, no one can prevent it or provide protection except Him. It's a powerful reminder for self-reflection and action. If we want better outcomes in our life, we must take responsibility for our own transformation.